Uh, our third speaker is Jenny Meinhardt. She's from Charité University and from Department of Neuropathology, and she's going to be talking about what SARS-CoV-2 does to our brains. It's a very important subject. Looking forward to your talk. Please.
Yes, thanks a lot. Hi, everyone. So yeah, I'm Jenny. I'm a neuropathologist, so not really a immunologist, but yeah, so probably my talk will be a little bit more pathology based. Yeah, let's see. So it's focused on brain alterations, so mainly alterations in the brain tissue during the SARS-CoV-2 infection. And as today's symposium is also called retrospective, I just wanted to give a short retrospective from our point of view. So for me and for us as neuropathologists, looking back at the pandemic and the last years, this time really also was kind of an opportunity to get closer together, because usually in Germany we have neuropathologists, pathologists, and forensic medicine. We are all doing autopsies, but traditionally we are working quite separate from one another. But during this pandemic, we somehow got closer together, and we managed to establish this national autopsy registry. The aim was basically to really systematically collect as many COVID-19 autopsy samples all across Germany. The funding period of the COVID autopsy registry ended in 22. Until then, I think about 34 centers participated with us, which is quite a lot for our field. And now we received another funding from the BMBF, and this whole concept is now being expanded to really other use cases way beyond COVID-19. So for us, looking back, yeah, this was kind of getting closer together all around Germany. And yeah, this was quite nice. And some of the data I will show later is also based on these collaborations. So yeah. Now back to what does SARS-CoV-2 do to our brains. So I think since the very beginning of the pandemic, these smell disturbances and taste disturbances, headaches, and so on have been described to occur during SARS-CoV-2 infection. And maybe the striking difference compared to other viruses is the amount of testing. So there have been a lot of diagnostic PCRs, so a lot of SARS-CoV-2 infections have been detected, even asymptomatic and mildly ones. And these neurological symptoms have been directly linked to this SARS-CoV-2 infection more often compared to other respiratory viruses in the past. But however, there have been the smell disturbances, the taste disturbances, and for us as neuropathologists, so in the beginning, the question came up, okay, does SARS-CoV-2 infect the brain? How might it enter the brain? And initially, also, our lab assumed that maybe SARS-CoV-2 might enter the brain via the olfactory nerve. So we did this project, and for us as neuropathologists, the gold standard for viral detection is electron microscopy. So we performed electron microscopy across different samples. And in this project, we were able to detect viral particles in the olfactory mucosa, but not in the CNS tissue. However, we had some PCR positive samples, and we also performed immunohistochemistry as shown in the picture. And we used, when we used this antibody, the SARS-CoV spike antibody that was available at that time on the market, we got this positive signal in brain endothelial cells. However, when looking back, I would say this has led to misinterpretations because, looking back, this antibody seems to bind non-specifically. beyond that, in our field, there was also the opinion that maybe there's no direct infection of the brain, but more like a bystander reaction of the brain to the systemic inflammation. And in this paper by Young et al., they haven't found SARS-CoV-2 in the brain. They described like an activation of brain barrier cells, in particular choroid plexus cells, due to the systemic inflammation. And this activation of brain barrier cells then leads to alterations of the surrounding brain resident cells. Yeah, and when you're looking through the literature of the past years, it's really that you will alternately find paper that either find SARS-CoV-2 in the brain or not. So this paper by Stein et al., they even described persistence of SARS-CoV-2 in the brain way beyond the acute infection. They performed some stainings, and they got a positivity, especially in cerebellum samples. So over the time, the literature got a little bit confusing, I would say, with really two opposing views, one finding SARS-CoV-2 in the brain, while others did not. And in this paper, this is kind of work of the consortium I mentioned in the beginning. Some colleagues of mine tried to establish clearer standards for immunohistochemical and ultrastructural analysis. I just put this photo here. It hangs in my colleague Carsten's office. So he has dealt a lot with misinterpretations of some viruses, vesicles as viruses. And he's always saying, Jenny, it's not looking like a sun or a ladybug. It's more a nutmeg. So keep in mind. So for us in the neuropathology, there were basically these three questions. Are there possibly entry routes to the brain, like the blood flow, or cranial nerves, like the olfactory nerve, or even the vagal nerve? Are we able to detect CNS cells that get infected by SARS-CoV-2, or is it more like a bystander reaction of the brain to the systemic inflammation? And in this study, we have used fresh frozen autopsy samples from the olfactory mucosa, olfactory bulb, human brainstem, and cerebellum. We divided the COVID cohort into two groups, acute and late. Acute meaning patients having infection less than 14 days, late meaning patients having infection for over 14 days. And we performed mass spec proteomics, single RNA sequencing, and spatial transcriptomics, and also some multiplex histology, but I will not show this today. So our first aim was, okay, can we find SARS-CoV transcripts in our RNA sequencing data set? So this is RNA sequencing data of the olfactory mucosa with this UMAP showing all the different cell types of the olfactory mucosa. And yes, as you can see on the right, we were able to detect some SARS-CoV-2 transcripts in different cell types of the olfactory mucosa, but not in any of our CNS samples. So this is the brainstem. We've also looked at the olfactory bulb and cerebellum, and we haven't found any SARS-CoV-2 transcripts in our data set. So we then looked deeper into the tissue, and as I've mentioned before, we've performed mass spec proteomics. So this is proteomics data of the human fresh frozen brainstem. The first circle is showing the comparison between acute COVID-19 cases and control. The second, the late cases versus the controls. Yellow is upregulation, blue is downregulation. So in yellow, we basically see an upregulation of different interferon-related proteins like STAT1, MX1, ISG15, and so on, especially in the acute disease phase and to a lesser extent in the late phase. I think this is as we would expect it. On the other hand, in blue, we see a downregulation of proteins that are related to synapses and synapse biology, especially synapse organization and presynaptic vesicle cycle. And this seems to appear in the acute phase, but also still in the late phase, the late cases. We've then had a look in the RNA sequencing data set again, tried to correlate the protein findings to the RNA data. And so here, this is brainstem data again. We calculated an interferon score. So this is basically a combination of interferon-related genes across the different cell types in the human brainstem. And you can see a slightly upregulation of these interferon-related genes, especially in the acute phase, again, in particular in cells of the vascular unit, some macrophages. And however, when looking at the far right, I also included these neurons here. So there seem to be a subset of neurons, we called them glutamatergic neurons, and they somehow show this upregulation of interferon-related genes in this acute phase. And as we are neuropathologists, we have been very interested in these neurons and ask ourselves, okay, where in the human brainstem are these neurons located that show these interferon upregulation? So they had like this upregulation of ISG15, for example. And this is why we applied spatial transcriptomics. So we basically cut the human brainstem, put the slice of tissue on the spatial transcriptomics slides. And then we looked for this gene signature of the glutamatergic neurons in this spatial data set. And the human brainstem really has this distinct anatomical regions. And for us, it was quite interesting that we found, so it's like the second row, we found this glutamatergic neurons to be mainly located in the brainstem nuclei of the vagal nerve, which we found quite interesting, as this nerve is really a long nerve going to the heart and gut of the patients, and also the ambiguous nucleus, which is the brainstem nuclei, which is mainly in contact also with the pharynx of the human. On the far right, there are activated microglia are shown, and they seem to be more evenly distributed throughout the whole sample. So yeah, so in this project, we had a look at very different cell types. I just put this summary here to keep it kind of short. So on the left, again, as I've mentioned, we had this neurons showing upregulation of interferon-related genes. They are mainly located in the brainstem nuclei of the vagal nerve, maybe pointing to the fact that this is like a sensing of the peripheral inflammation via this nerve. On the other hand, we also had a look at endothelial cells. So when you look at endothelial cells, we saw an activation of endothelial cells, and yeah, as described in many other papers, we also found an impairment of the blood-brain barrier, microglia, astrocytes. They showed reactive phenotypes, and we also found a subset of oligodendrocytes to be reduced in their quantity. So overall, our data point to the fact that there's no real infection of really brain resident cells by the virus, and that it's more like a bystander reaction of the brain to the systemic inflammation, mediated via the bloodstream and the blood-brain barrier, and perhaps also via this granulate nerves like the vagal nerve. Yeah, so our data is basically all based on autopsy tissue. So these are all really severely affected patients lying on ICU, dying, getting an autopsy. Yeah, just to keep in mind. The last slides are just a short outlook what we are doing right now. So as many other people, we are also interested in finding alterations in post-COVID-19 condition. As there's no autopsy tissue from these patients, right now we are using cerebrospinal fluid samples, and we are comparing them to acute COVID-19 cases and controls. When looking at the CSF routine parameter, like cell count, glucose, lactate, protein, they seem to be rather normal. So we've also performed mass spec proteomics on this samples. And this is really just preliminary data with a small set of samples. We are currently reanalyzing it, but we just found it quite interesting. So when you look at the left, at the PCA analysis, and also at the heat map, maybe at the top of the heat map where the dendrogram is located, you basically see a quite nice separation of these three groups. So in red and orange, there's the post-COVID-19 samples, in green, the controls, and in blue, the acute COVIDs. So the proteomic profile of the CSF somehow seems to lead to a distinct grouping of these samples. We are currently taking a deeper look at these proteins. We've just performed a quick pathway analysis on this. So this is basically showing post-COVID-19 versus controls. Again, yellow is upregulation and blue is downregulation. And we see somehow like an upregulation of pathways related to the innate immune system, and also some pathways related to B-cell receptor activation, B-cell receptor regulation, B-cell receptor signaling to be upregulated in these post-COVID-19 patients. When looking at the proteins, this is mainly due to an upregulation of immunoglobulin light chains. We don't know what this means, so if anyone has an idea. And in blue, we basically see a downregulation of proteins, like in the corner, neurexins and neuroligens. So again, proteins that are associated with neurons and synapses. Yeah, but this data really has to be put in context and further analyzed. Yeah, so over the past years, lots of people have been involved. I just want to thank everyone, and yeah, vielen Dank.
Thank you so much, Jenny. It's a beautiful talk, and I'm sure you will get more results after you complete your study, hopefully. Yes, please. Winfried.
Thank you very much. This is very interesting. So I was wondering, your last piece of data on the CFS samples, do you actually regularly find immune cells, lymphocytes in the CFS samples of patients?
So it's
to explain actually why the B-cell signature comes from and so on.
So there are really few lymphocytes in it, and there are not more lymphocytes compared to the controls. So this is quite, we don't know where this comes from. So there are not more B cells in the post-COVID patients compared to the controls. Yeah.
Thank you very much to this insight into the neurological aspects and the NATON initiative that I think really is a changing system within NOM that might be really relevant. One of those that are relevant for the future. These might be cases that were heavily infected and severely sort of died due to COVID-19 before vaccination. And my question to you would be, do you stratify all your patients in your neural analysis, but also in your long COVID analysis due to infection before vaccination and after, and especially in the post-COVID or long COVID cohort, that might be relevant because the early ones that were long COVID before vaccination was around might differ also immunologically from those that have been vaccinated and then developed into long COVID syndromes. So do you do that also, and do you expect differences in your proteome analysis?
Yeah, so the first data really was just as brain tissue from the first wave. So wild type SARS, no vaccination. We haven't looked at any other variants on the tissue level. For the proteomic data, we are dividing the groups, but we haven't analyzed it yet. We are still sampling, taking samples, so it's still ongoing. Yeah, we will look at that and keep it in mind, but no data yet.
Please.
I have one question with respect to autoantibodies. Probably also analyze the sera of these patients, the severe cases of COVID-19 and long COVID. Is there anything known about autoantibodies against, particularly against G protein coupled receptors or neuroreceptors? Do the tissues stain for antibodies? I mean, are the neuronal cells stained on the surface for antibodies?
So we have not done it, but I think some colleagues from the Charité have looked at antibodies and found some autoantibodies in the post-COVID patients against some neuronal proteins. But
in the serum.
In the serum.
But the brain, the stem.
We haven't checked.
Histology doesn't change. Show any changes.
Oh, I haven't done it. So.
There is one more question from back, Andreas, on your left. Yeah. Yes, please. Yeah, you have a microphone.
Yeah, thank you. I actually have a couple of questions, if I may. So the neurological symptoms that can range from mild cognitive impairment to severe encephalitis. So my first question would be, do you suggest that there is a common etiology, so bystander activation, rather than infection that is just has different levels of severity? And did you see any, or can you comment on risk factors for this enhanced bystander activation in the patients that you saw? And then lastly, in the beginning, you alluded to us having a lot more information on COVID compared to other viral infections that also come with neurological symptoms. So do you think that we can learn something from SARS-CoV-2 with respect to other viruses, such as measles, or would you think that the etiology is different since the virus is different? So is there anything we can learn from COVID?
Yeah, I think we can learn from COVID, maybe comparing it to similar or also other respiratory viruses, maybe not so different viruses, but maybe we could compare the reaction to a severe influenza, maybe. We haven't looked at influenza tissue on this level right now, but I think it's not specific for a SARS-CoV-2 infection and could be transmitted also to other respiratory or heavy respiratory infections. I think I forgot the first question. Sorry.
Yeah, so do you think that the etiology of, let's say, a mild cognitive impairment and encephalitis is the same, so bystander activation, or would you see those as separate neurological conditions?
Good question. So our samples are really just heavily affected patients. We haven't seen encephalitis in our samples. And it's really quite a distinct cohort, mainly male, first wave, wild type SARS, lying on ICU. So yeah, I think all the other samples have to be analyzed. Probably there's much in common. Yeah.
Thank you. The last question was about risk factors. So was there anything in your patient population that stood out in terms of immune deficiency or any other risk factors you could speak to?
No, I would say the known risk factors that have been known in the beginning of the pandemic. So male, elderly patients having comorbidities like diabetes, hypertension, and all this stuff. But we haven't looked really at immunodeficiencies. Yeah.
Okay, thank you so much,
you.
Jenny, again.